Hello, this is Dr. Dan Guerra from VerEvMed's studios in the Pacific Northwest. Today I'm going to talk to you about ethanol and its opportunistic cause of neural damage and deficits associated with neuropsychiatric symptoms. Obviously, that's a huge topic, and I'm only going to talk about a very small segment of it, sort of like as a way to segue into it so that we can look at the literature amongst multiple lines of evidence. So today, I just want to sort of introduce this subject. Now, I did previously introduce it briefly when we were talking about cannabis and its effect on the central nervous system. So today, the front and center is going to be ethanol. Now, most of you know that ethanol is well studied. Its intoxication and its effects on intoxication, as well as on neurochemistry and neurophysiology and pathophysiology in humans relative to, say, the liver, the kidney, the heart, muscle tissue, bone structure, you name it. Because we have a National Institutes of Health that is exactly about alcohol abuse. So as you can guess, we know a lot more about ethanol in humans than we did about cannabis. But in my previous lecture, the one I just gave a few days ago, I emphasized how we need to do more studies in cannabis, but it doesn't mean that we should stop working on ethanol, because there's a lot of things we're still learning, especially crosstalk with this toxin and other toxins. So let's get started. Okay, so you all know by now that this is VerEvMed, and that's me. I'm Dr. Dan Guerra, and I'm co-founder and chief science officer of my company. That's my particular and significantly individual email address, as far as I know. You can contact me at that site if you want to talk to me about things in these lectures that I give, or if you want to hook up with our company. Now, our company's website, I guess I didn't put it right here, it'll be at the end. It's just www.verevmed.com, but you can contact us directly through there. There's an email address there. You can certainly enter into our services that way. Remember, these are free lectures that I'm doing on YouTube video just to describe the kinds of things that we do, the in-depth evidence discussion in the primary scientific literature. All right, and that is the Facebook page down there at the bottom, so go right to that and you'll see all of these videos as well as lots of other really interesting things. Let me get rid of that. Okay, so you're looking deep into the forest. This happens to actually be in Colorado at about 9,000, no, actually about 10,000 feet, and near Monarch Pass that we were at just last summer. Anyway, I'm showing you forest pictures because, of course, I love the forest. I'm all about going to the woods all the time, but also because I want you to get the idea that we're just walking into the deep part of the forest, and we're just entering it in terms of the effects of ethanol on human biology, basically. It's a tremendous subject, as I've already said, and we're only going to talk about a very small segment of it, and we're going to look at some literature. So let's get started. Yeah. All right. This comes from the National Institutes of Health, the alcohol abuse division of that. On the right, you see your typical neuron, right, with cell body and with the axon and with dendrites, right, the terminal butons there that are going to be adhering to the next adjacent neuron, right? Now, the point I want to make here, today we're going to be talking about myelinated neurons, okay? It's a very specific subset. And we're going to talk then about this open area of the axon and then the areas that are insulated by the myelin sheath, okay? Now, you've probably heard about this before. Certainly you in the medical profession know all about this because there are various diseases that are demyelinating. MS is one of the major ones I can think of. And the cells that make up the myelin sheath are variable, but the major one is oligodendrocyte. And the oligodendrocytes then, somehow those are removed or during demyelination, or they're degraded. And that degradation process then removes that insulation, and then that corrupts neural transmission where the myelin sheath is covering those. Now, not all axons are myelinated. I think most of you know that as well. And if you don't, now you know. But that myelin aids in the transduction, or specifically the saltation of the action potential down that axon. So it makes basically the neural signal move much more rapidly. And the other thing just right now to keep in mind, because I called it an insulator, it's more than that, but basically it is an insulator because it's composed of a large amount of lipid, and particularly sphingolipid, although there are sterol-lipids as well, as we'll see. So anyways, if you lose the myelin sheath, there are various diseases, and one of them is MS, and it can end up crippling a person or killing you, of course. But also there are processes in the body which allow for remyelination. So it means that there's a potential out there for us to be able to reconstruct that myelin sheath by pharmacotherapy and maybe by other kinds of therapeutic means. But point here is that chronic alcohol consumption causes a shrinkage of brain tissue. These are just some of the things that happen. A degradation of the fibers. Now, by the fibers, I mean the axons, and when accompanied the myelin sheath. So that's known as white matter, because that's what it looks at under certain phase contrast microscopy. At any rate, those fibers, it's a degradation of those fibers that carry information between the brain cells. And the brain cells, or the actual neurons in the glia themselves, of course, are the gray matter, right? That's what they mean by that. Just again, because of what it looks like under microscopy. Now, the brain's gray matter, as I said, consists of neurons and the surrounding glia. White matter is made up of the axon. Okay, that's the long extension of the neuron, as you see here. All right, now some white matter, not all, is surrounded by myelin, and I already started to introduce that. The axons form fiber tracts, then they're called, and they link nearby and distant neurons across different brain regions. So you have what are known as white matter tracts, and this is the target. This is what gets damaged by chronic alcohol consumption. Okay, now put on the very nice spectacles here. And I'm going to go through very quickly. This is just a paper showing you basically the composition of myelin versus gray matter versus straight white matter. And the composition, by that I just mean macromolecules like protein, lipid. And you can see that there's some difference between human myelin and human white matter, right? Some difference in protein content. You can see that myelin has a lot more lipid, for example, than the rest of the white matter. That would be the axon. And that it's also quite different, of course, from the gray matter, okay? And then if you look at whole brain, all of that fine detail molecularity of that data gets confused up because you're just looking at whole brain. Now, that happens to be rat. I didn't extend this particular table, but we also can look at whole human brain as well. But the important point here is look at all of these lipids: galactocerebrosides, sulfatides, total galactolipid. You can see here there's quite a difference or distribution, a variation. Look at how low it is of galactolipid in gray matter as opposed to myelin, as opposed to the white matter, okay, in humans, for example. So these are all basically sphingolipids, a topic I had quite extensively covered in the last several weeks in dead winter. I'm so happy. It's like March already, so pretty soon we're going to be in springtime. Yippee. Anyway, all those lipids, you notice there's a significant component of sterol, cholesterol. There it is right there, right? So there's a lot of cholesterol in this lipid layer as well. And it's kind of uniform, so you don't see a big difference there. But I want to point out that there are sterile lipids. So lipids which are sterilated, that is, they have a cholesterol component, but that's not all they are. And then you can see there's a lot of glycerolipids. Here is a plasmalogen, that's ethanolamine phosphatides, that's a plasmalogen, ether-linked lipid. And then you see your standard typical glycerol phospholipids, phosphatidylcholine. They're down there as sphingomyelin. You can see the sphingomyelin a fair amount in human myelin, as you might guess. There's a fair amount in total white matter because it's included in that. And you can see that even gray matter has some sphingomyelin. So I don't want you to think that sphingomyelin is only in those white tracts. It's also in the rest of the central nervous system and the peripheral nervous system too. PI, phosphatidylinositol, is always low. It weighs in at a very low molecular amount. However, it's really important because PI is a signaling lipid. And again, we talked about that. All right, so that just gives you an idea of the distribution of lipids. The reason I bring that up is because there's a lot that ethanol does that corrupts lipid metabolism. So as a lipid biochemist, we're aware of this, and that's the kind of thing that we consult on, is how ethanol, that particular drug, that toxin, affects lipid metabolism. It affects it in many different ways. All right, here's the paper we're going to actually be looking at. It came out in December of 2017, so this is March 2018, so only a couple months ago. And what I want you to do is, first of all, remember that white matter are these tracks, okay, that you see and this is transverse section here of a brain, a transverse section. So white matter are bundles of millions of nerve fibers, axons. They carry signals from the neurons, one part of the brain to the other. Don't worry about the rest of that. It's very popular press in a flash like cables. Okay. And the gray matter, as I said, are the neurons in the actual glia. There's a sagittal section here showing you what it looks like. There's the gray matter. You can see it's darker, and the white matter is lighter. Okay, and you can see this throughout these different scans. So let's talk about it now. Chronic ethanol intoxication results in white matter loss. Goodbye, glasses. With the cognitive impairment characterized mainly by loss of executive functions. By that I mean the processes that are believed to be controlled executively by the prefrontal cortex. Of course, that can lead, chronic ethanol ingestion, that is, can lead to dementia and disability and death. Okay? Neuroimaging and post-mortem evidence, we have a lot of it, demonstrates white matter atrophy and neurodegeneration correlated with maximum daily and lifetime alcohol abuse. So that is, the more alcohol you consume, the more white matter atrophy, the more neurodegeneration, the more behavioral impact, and the more disease. Okay, neurodegeneration, for example. In fact, we actually have something called alcohol-related brain degeneration, which is a syndrome. Okay, ARBD. And it's prominent in certain regions of the brain, and I talked about them last time when we talked about cannabis, so I'm just going to mention them here. Corpus callosum. Now, the corpus callosum is basically in midbrain, and it basically is how one hemisphere of the brain talks to the other hemisphere of the brain. So you can imagine that's really important. In fact, the corpus callosum is also where epilepsy is sort of, that location of the brain is where epilepsy derives its corruptive behavioral patterns. The seizure. All right, so there's that. And then you have the prefrontal, temporal, and cerebellar white matter. Okay, so those are the regions that we're talking about getting destroyed, basically, by chronic ethanol use. Degeneration of the corpus callosum disrupts, as I just said, the that means both parts, hemispheric communication, including sensory, motor, and cognitive information. So obviously that's really a bad thing, right? And we know this, when a person's inebriated, when they're drunk, when they've been intoxicated sufficiently with ethanol, you notice that they have problems with their sensoria, they have problems with motor neurons, and they certainly have a problem with cognitive information. In fact, many crimes, violent crimes, are caused by people who are under the influence of ethanol. Okay, and this is well known by the police. All right. Degeneration of the corpus callosum disrupts we already said that. So ARBD associated white matter atrophy is characterized by demyelination, which I just showed you, dysmyelination, which means a corruption of the lipid metabolism per se, and axonal degeneration. So not just the myelin, but the axon itself. Okay. So in order for you to get an idea of how this is studied, using scanning. This is a great thing about scanning. Of course, people, I think, abuse scanning because they think they can get a lot more information than I believe they really do. But basically, you can look at oxygen use by scanning, by functional MRI, for example. But you can also get a real good idea of some of the structures in the brain. So perfectly, healthy person, non-invasive scan can give you a lot of information. In fact, you can look at an alcoholic brain and compare it to a non-alcoholic brain, and whether or not the person denies they use alcohol, you'll be able to pretty much diagnose that they do, and not only use it but abuse it, which is probably the same thing, at least in my perspective. Now, okay, so there are a couple of things to consider here in these diagram here. You have isotropic or isodiametric, and you have anisotropic diffusion. Here we're just talking about bulk water. So you can use, for example, deuterated water and watch water move through these tracks, okay? So water molecules in the brain are constantly in motion, so that helps you be able to get a scan from them, and that's called Brownian motion, of course. The motion is unconstrained, as in the large fluid spaces called the ventricles, okay? And the diffusion is isotropic. As I said, it's isodiametric. It generates a field which doesn't have any particular bias in the 4 points of these axes, of this XY Cartesian grid, right? All right. That means that motion occurs equally and probably randomly in all directions. I say probably randomly. I doubt that's really the case. Nothing is random in biology. It's just pseudo-random, I think. But when motion is constrained, such as in these tracks, as in white matter tracks, yep, illustrated here, diffusion becomes anisotropic. That means it no longer is isodiametric. So the motion is oriented in one or more direction along one of these axes. Okay? So that lets you, it gives the ability of the scanner, the person who's analyzing the scan, to know where those white tracks are. Okay. So once again, we're looking at transverse section here on the right. And okay, so we're looking in coronal, these are all coronal orientation from MRI and DTI studies of a 61-year-old healthy man, which are the upper images, and a 60-year-old alcoholic. It's the same person. So this is a healthy man who's 61. This is a man who's very close to age 60, who is an alcoholic. Now you can just tell right away from the MRI, there's a tremendous difference in these scans, right? Look at the corpus callosum here versus there. So the MRI of the thinner corpus callosum is displaced upward by enlarged ventricles, okay? And on the DTI, it's a different type of scan, which stands for diffusion tensor imaging, and it's particularly useful for looking at white matter. That's why it's done. You can see, you strip away, you're not looking at any of that gray matter, it just gives you a dark field, right? Yep. The less well delineated white matter tracks in the alcoholic man, see how you see a lot of like fuzziness around here, as compared to the healthy man where you see much more sharper line of track. That tells you there's a problem with myelination and with axonal degeneration. That's what it tells you. You see, like I said, put a person in a scanner and they swear up and down that they don't drink alcohol, even though you might smell it on their breath or something. And then you look at their scan and you go, yeah, well, maybe that's not quite true. So scans are pretty amazing. That particular element of it is anyway. All right, so let's talk a little bit more about what ethanol does. Again, this is by no means an exhaustive discussion. In fact, I'm not really even going to talk about what causes intoxication with ethanol. That's another entirely really fascinating biochemical story. But and when I teach biochemistry, I do always discuss this, particularly around mid-March, and you know why? Because that's when you get close to St. Patrick's Day, which seems to be like the holiday for boozing it up. I try to tell the students, maybe consider like checking that alcohol abuse at the door, and not only the designated driver, maybe everybody should just take an alcohol like holiday. In fact, maybe an alcohol holiday for the rest of their life. Just an opinion of mine. At any rate, alcohol, metabolism, biochemical impact, just very briefly. Ethanol more would resemble carbohydrate, but it's not really, of course. But it's metabolized like a fatty acid. That's because it's about oxidative reduction reactions. The alcohol dehydrogenase enzyme itself, you have ADH in liver cells, and you have this NAD requiring dehydrogenase. Okay, that's what it is. It requires nicotinamide adenine dinucleotide. So that's really important right at the get-go, because NAD, NADH, that couple, is central, dogmatic, absolutely vital in all of metabolism. The ratio of NAD over NADH or vice versa it's absolutely tantamount of which way metabolism goes. Oxidative or reductive in terms of biosynthetic, degradative, lipogenic, or gluconeogenic, or indeed even controlling things like DNA synthesis and protein synthesis. It's very important. And even things like chromatin condensation, because the sirtuins are NAD requiring, not NADH. The sirtuins being the deacetylases, which control that aspect of chromatin utilization for remodeling. So it's very, very important, just that one molecule. So there's also a microsomal, that means ER, ethanol oxidizing enzyme. It's called the MeOs, and it uses cytochrome P450 enzymes. And basically it uses an electron transport chain that's embedded in the ER, okay, and particularly in the smooth ER. And so tolerance, ethanol induces synthesis of those enzymes. So what that means, you have your normal alcohol dehydrogenase, that's your normal way of detoxing this terrible toxin you just took in your body, this ethanol, which has no business being there. And so you have alcohol dehydrogenase. If you have adequate amount of that enzyme activity coming from the liver, you might be able to detox it, but you're not really over yet, because then you make the next degradation product, which is acetaldehyde, which is extremely toxic. So you have to really take that all the way to acetate, as we'll show in a moment. But anyway, what can happen when you get an overload, that is not a chronic ingestion of ethanol, but some kind of bombastic overdose of ethanol, right, like a binge drinking, then enzymes are actually turned on in the microsomes, in the ER, that give you an alternative way of metabolizing it. That is getting rid of or eliminating the toxin. That's the point there. Here are some of the reactions then. Ethanol with alcohol dehydrogenase going to acetaldehyde. Notice that you're making NADH when you do that reaction. And the reaction is really reversible. Although, going in this particular way, going from acetate to ethanol, that's alcoholic fermentation. And that doesn't really happen in humans. That happens, of course, when you add yeast, for example, to plant extracts, you'll go through an ethanolic fermentation. That's how they make ethanol. That's how you make the amount of ethanol that's made, for example, in wine. Then you distill that. And when they distill that, means that they increase the amount of ethanol in concentration by carrying out a distillation. And that's how you make all of the the hard liquors, right? Whiskey and vodka and gin, etc. Of course, all of you probably know that. All right. So now, when you metabolize ethanol, though, you make acetaldehyde, a very dangerous, very toxic substance. Okay. Back with the glasses. Notice with contrast, I always need the glasses. It's strange. So acetaldehyde itself, super bad intermediate, because it will make an adduct to DNA, RNA, and protein. When it chemically bonds to DNA, RNA, or protein, that's never a good thing, because you're not supposed to have acetaldehyde as an aldehyde anyway, which has its own problems as an aldehyde. But when it binds to DNA, RNA, or protein, it causes those three macromolecules of central importance in life to dysfunction. You also get increased reactive oxygen. Because of these reactions. That increased reactive oxygen, of course, will cause damage to DNA, RNA, protein, and lipids. And also, you get this massive whopping increase in this ratio of NADH and NAD. And that, of course, will shut down, as all of you who are good card-carrying biochemists know, once you shut down the TCA flux, you just shuttle all that citric acid back into the cytosol. You lyse that citric acid back to OAA and acetic acid. You make acetyl-CoA, actually. Acetyl-CoA is on its way for lipogenesis. That's why when you ingest ethanol, you make fat, basically. Okay, that's what's shown here. All right, there's your type 2 enzyme using now NADPH and molecular oxygen. And that's, again, happening in the microsomes. All right. So let's take a look at this again in more diagrammatical sense. There's alcohol. It's being metabolized to acetaldehyde, makes these adducts. Bad news. You get oxidative stress because of reactive oxygen being made. If it reacts with lipid molecules, you get then sort of a chain reaction of lipid peroxidation. And that lipid peroxidation includes malinaldehyde adducts, which will bind to proteins. You also add acetaldehyde here, we just talked about, making the acetaldehyde adducts and the MDA adducts. This is all bad because these proteins are no longer functional like they normally are. This is a 9-carbon compound that binds to proteins after lipid peroxidation, because if you have an 18-carbon fatty acid and you split it in the middle because of peroxidation, you get 2 9-carbon compounds and acyllipids. And when you make that, you also get protein adducts. And the same thing here with the hydroxyethyl radicals with proteins. So all of these products are dead end and pathogenic in the cell. So there's your cytochrome P4. So even when you metabolize the alcohol, which is what your body's trying to do, because the alcohol itself is a toxin, okay? So your liver recognizes the alcohol concentration like no other toxin and tries to eliminate it. But during the elimination process itself, you make all of these other even end products that are even sometimes more dangerous than the original drug, okay? So that's the point you have to understand. That metabolizing alcohol is not enough. You are causing damage downstream from the ingestion of the alcohol, which can last far longer than during the time of intoxication. In fact, chronic use of alcohol can corrupt a person's entire lifespan. So, once again, we know that people who have a problem with controlling their alcohol intake, we know there's a lot of people like that worldwide because alcohol is a very addictive drug. Because again, it impacts the reward pathway. I'm not going to talk again about how alcohol makes a person feel inebriated this time, but I will in some other subsequent VerEvMed lecture. Anyways, there's alcoholism. We know it's out there. That's why we have a National Institute of Health about it. And there are biochemical and there are overall just metabolic alterations. First of all, you have the acid-aldehyde toxicity. I just told you made those adducts. When you have this high ratio, what that causes is you synthesize more lipids. So that can cause obesity for people who consume a lot of alcohol. And I think we know a lot of people who are alcoholic, at least the early stages of it, tend to gain a lot of weight. They tend to be puffy. That has to do also with a lot of extravasation of water because of membrane destruction. But we also know basically that, that there are a lot of what they used to call, in parentheses, fat, happy drunks, right? And only later on do they lose weight, and it's usually right before death. Okay, there's also a lot of substrate competition for the reactions for alcohol metabolism. And there's also an induced metabolic tolerance. So we know this, like a lot of addictive drugs, the more alcohol a person consumes, they get a certain tolerance where it takes more and more of the ethanol to get them high, basically. And that doesn't mean that there's less intoxication to the organs or to the metabolic system or to the brain. In fact, it just means that that happens more. You get more of this onerous destruction of central nervous tissue, neurons and glia and axons and myelin sheath because you're ingesting more alcohol, because the way the addiction works, you are able to process it. You get a metabolic tolerance. None of that is good. Because that means the chronic use of alcohol. People don't just have one glass of wine, but then they start having 2 glasses, 3 glasses every night, and that leads to, even though they might not think about it, basically chronic alcohol abuse. You put a whole life behind that, and you get all kinds of metabolic and therefore health issues, probably in the billions of dollars, let alone all the crime that's caused by alcohol consumption. All right, so here are the reactions. There's ethanol, there's alcohol dehydrogenase, there's acid-aldehyde dehydrogenase. I warned you you'd see it. And there's making acetate, okay? So this is, acid-aldehyde is toxic. Ethanol is toxic. Acetate alone is not, but it has to be metabolized. And the way it's metabolized, it usually basically just gets condensed after it gets esterified to coenzyme A and make a thioester with it. You condense it then with oxaloacetic acid. You make citrate. Again, because of this high levels of NADH you're making, it's a very reducing environment in this world. You shut down the TCA cycle. You shut down all the dehydrogenases in the cytoplasm as well, which I'll show you in a minute. And basically what you do is you make fat. You make lipid, because that acetate thing just becomes a lipogenic substrate. Now, notice that methanol is also converted fine by the ADH aldehyde dehydrogenase reactions. And this is why methanol is so darn toxic, because it makes formaldehyde. That's how people go blind and have acute toxic liver failure by consuming methanol. And there's all kinds of horror stories in the past where people drank methanol and died, right? And formic acid as well is not a very good thing to be synthesizing in the gut or have in the blood. Okay, so ethanol is not the only reactant, is what we're saying there. Here's lactic acid synthesis. Now, here's glyceraldehyde 3-phosphate. This is just part of what glycolysis, of course, is glyceraldehyde 3-phosphate dehydrogenase. Normally, it's making NADH in glycolysis. That gives you pyruvate several steps later after you go through 3-PGA, 2-PGA, and PEP. You make pyruvate. Then the lactate dehydrogenase uses the NADH normally from the GAP-DH reaction to synthesize lactate. And that's exactly what happens. The more NADH you're pumping into this system, the more lactate you're making, okay? And the more lactate and also the more lipids. So that's another standard thing. You have lactate acidosis with a chronic alcohol abuser, and you have increased lipogenesis, and that's where it comes from. Okay? Simply because of this ratio of NADH to NAD, cytoplasmically as well as in the mitochondria. All right. Now, just looking at the pyruvate dehydrogenase couple, you know that pyruvate dehydrogenase does make NADH, and this is how you make basically acetyl-CoA. So there's the final reactant there. You bring in pyruvate. You go through 5 reactions using 3 proteins in the PDH enzyme. I love teaching the PDH enzyme biochemistry because it shows you a lot about structural and functional subunit interactions to make a polyprotein to turn into one enzymatic reaction. And all the enzymes stay on the enzyme surface anyways. Sorry. But it's just a beautiful thing. Reduction of sulfhydryl groups and disulfide bonds being formed transiently. It's just a wonderful enzymatic machinery. The point I'm trying to make here, though, is that the product is NADH. So if you have a lot of NADH, what's going to happen to this enzyme? You're going to start intoxicating this enzyme, okay? And so what happens to the pyruvate? It makes lactate. You see how this works? Okay. You also have the Malic enzyme. Most people don't consider this, but I'm a biochemist, and I love the Malic enzyme as well. So what does the Malic enzyme do? Basically, pyruvate carboxylase. So this is like a gluconeogenesis series of reactions, making OAA. Now notice here it uses NADH and it makes malate, okay? And then malate can be reconverted to pyruvate, okay, by decarboxylating reaction that also makes NADPH. Malic enzyme is really important because it's a cytosolic source of NADPH. Why is that important? Because NADPH is necessary for lipogenesis, in fact, for anabolic biosynthesis in general. And with the oxidative pentose phosphate shunt and the Malic enzyme, this is where you get the cell's reserves for NADPH for reductive biosynthesis, such as in lipid biosynthesis. So you know that if you run through the pyruvate dehydrogenase reaction, you make acetyl-CoA, though. That happens at the mitochondria level. In fact, it occurs in the mitochondria. And then you get this condensation reaction, making citrate. High levels of NADH again in this system is going to pump that citrate out, and you're going to make fat, right? So that's the point. Now, notice that when citrate is cleaved, the acetyl-CoA can go on to make lipid, but you have OAA. And the OAA can go on to make malate, and malate to pyruvate. You see here, this is exactly what we're looking at, this couple down here, right? And then you can send pyruvate back out into the cytoplasm. So this Malic enzyme, this whole process here, is coupled to what happens when you get a toxic ingestion of ethanol, right? You're driving these reactions, basically running the carbon into making lipid and lactic acid. Right? And you're not really getting anything else out of it. It's not a good metabolic end note or fate. Now, let's talk a little bit now about ethanol and neuropathology. Okay, so we'll take a look at this section here in a second. This comes from a paper published quite a while ago in the Journal of Biological Chemistry, my favorite journal of all. This is showing you tenascin R plays a role in neuroprotection via its distinct domains that coordinate to modulate microglia. Now notice, just look at this and then we'll do all this verbiage over here, that this one protein, this one protein has different functional domains. And depending on how many of these repeats there are, you're either going to turn on protein kinase A or protein kinase C. Now look, protein kinase A inhibits microglial adhesion and migration in the brain. These are brain resident macrophages that we talked about in the cannabis talk just a couple of days ago. But notice that if you have enough of these repeats and they're expressed, that'll turn on protein kinase C, which does the exact 180-degree opposite. It'll cause adhesion of microglia and migration. Microglia, again, depending on whether or not they are induced to be activated, can then start synthesizing pro-inflammatory cytokines. So that can induce neurite outgrowth, kind of like what we're talking about, that arborization. And that arborization is one of the ways you get intoxication not only from ethanol, but also, as we saw, from tetrahydrocannabinol. Okay, so this is embedded in the previous talk I gave, but it's also giving you the basic understanding of what proteins are involved. And just love biochemistry. Okay, let's go over here. So there's something called the Wernicke-Korsakoff syndrome, and that's basically dementia, ethanol-induced dementia. Okay, there's a clear association between the reduction of thiamine levels or thiamine-mediated enzyme activity. One of the enzymes is the one in the oxidative pentose phosphate shunt, transketolase. Okay, so and also thiamine pyrophosphate is utilized in amino acid metabolism. All right, so very important central stuff. It's been established, or this has not been conclusively established, is that in the case of peripheral neuropathy, certainly central neuropathy. So the effect of thiamine, that's why chronic alcoholics, you look for thiamine deficiency, and immediately when they're taken off the booze and they're detoxed, they put on thiamine therapy. That's one of the reasons, okay? Now, in a comparison of a patient with alcoholism and the non-alcoholic control, it was concluded that nutritional deficiencies alone did not produce all that neuropathy. So it's something beyond just the thiamine. It's like, of course it is. I just told you that ethanol is like a major central nervous system, peripheral nervous system, metabolic, whole organ toxin. So it's not just going to be affecting thiamine. Bad enough, right? Because thiamine controls so many frank, potent enzymatic pathways in the cell. Corrupting that is bad enough. But the neuropathy alone is not understood or obtained just by thiamine deficiency, which is induced here. So ethanol is toxic to the autonomic and peripheral nerves in a dose-dependent manner. This we know from animal studies, based on heart rate, blood pressure, and all the electrophysiological examinations also in humans, right? It's independent of thiamine deficiency, so ethanol now appears to have a direct toxic effect on peripheral nerves. Again, this is now chapter and verse of what we know, but when we first started studying ethanol, we didn't know this, right? This is something that came about after doing a lot of really good research, NIH coming through the funding and good research on ethanol intoxication, of course. But also, NIH isn't the only organ that funds ethanol research. There are also pharmaceutical companies that do, because they're making drugs that try to block the various pathways that lead to, for example, adduct formation, not addict formation. All right, perhaps catecholamines. I told you this is a reward pathway, dopamine and catecholamine. Perhaps catecholamines in nociceptors, those are receptors that receive their pain signaling, output as pain signaling, are metabolized to neurotoxic products by the monoamine oxidase system, which of course does happen. And that can cause neuronal dysfunction, which can lead to neuropathic pain. So one of the things in the Wernicke-Korsakoff syndrome is a lot of pain in the peripheral nerves. Okay? Painful alcoholic polyneuropathy with predominant small fiber loss and normal thiamine status is well known. So it's here, you're getting all this polyneuropathy, you're getting a lot of pain, you're getting all these neural tract degeneration, but it's not thiamine associated. So it looks like there's a direct neurotoxic effect by alcohol or its metabolites. In fact, exonal transport and cytoskeletal properties are impaired by ethanol exposure in animal systems. And this is finally why I showed you this diagram. pKa and pKc may also play a role in the pathogenesis, especially associated with painful symptoms. This has to do, again, with the activation of microglia, generating cytokines. You also get the synthesis of oxylipids, and oxylipids cause vasoconstriction, and that causes pain. Okay, so there you go. Gut pain, too, for example, from acute ethanol ingestion. It works the same way. So acute and chronic pain syndromes are usually categorized into inflammatory and neuropathic based on their etiology, obviously. And this incurs both, alcohol does. For musculoskeletal pain, however, the distinction has not been so clearly established because they overlap, even though muscle pain is observed in neuropathic as well as inflammatory. So that's the point I'm making. Ethanol causes both kinds of pain, right? A direct and pro-inflammatory one, and this neuropathic one as well because of its effect on membranes. All right. Paper published about now 5 years ago, not quite actually, late November, so that would have been 4 years and a couple of months ago. Ethanol produces a wide range of pharmacological effects on the central nervous system, ranging from anxiolytic effects. Of course, that's why people drink it. the neuropsychiatric use of the term anxiety, right? Having like an uneasiness of everything unknown on the day-to-day basis. Alcohol, definitely people do self-administer alcohol, that is, they drink alcohol to release their tensions, right? So that's one of the reasons that people do it, because of this direct effect on the central nervous system. But of course, that's also what leads to intoxication and alcohol addiction, right? Because of turning on that reward pathway, people feel at ease and they want more and more alcohol because it makes them feel at ease all the time, right? Whiskey River, take me home. Not much is known about the molecular mechanisms, however, even though we've been studying it for a long time, the interaction with specific target proteins such as ion channels. Okay, so this paper was saying, preceding the National Academy of Sciences, saying even like as late as 2013, late 2013, what about ion channels? Okay, it's just another molecular target we're looking at here, okay? Because we know that ion channels are important in this whole induction of intoxication, okay? All right, now check this out. You have these G protein, that means they bind GTP, gated inwardly rectifying, or GERK or KIR3 channels, right? Inwardly rectifying, bring potassium in. And they're activated by concentrations of ethanol that are relevant to human consumption. If you want to know how much that is, 18 millimolar ethanol in the blood is also a blood alcohol content, or BAC, or here is called blood alcohol level in Britain, of 0.08%, okay, have been found to play a key role in alcohol-related disorders. Gee, isn't that interesting? Here's another target for alcohol. That wasn't enough what we already covered. For example, this is now looking at mice, where we can knock out genes, and then they ask, what effect does it have? When you knock out a gene, you can say, if the gene's not there, what effect does that have on normal physiology or pathophysiology? Okay. Now, there's a lot of caveats to that kind of research, and I'll be the first one to mention that, because way back when, in my earlier days at the bench, I was working on homologous recombination, gene knockout. And so we used to think that gene knockouts would really tell us discretely and absolutely specifically what each gene was doing. And that was not really what most of us believed, because we knew that genes interact with one another, right? And as gene products interact with one another, DNA interacts with one another, RNA interacts with one another, and all of metabolism causes everything to be completely dynamic and completely complex and uncertain, right? Because an open system. All right. Sorry for all that, but I'm sure that that was important for you to get. For example, mice lacking the GERK2, okay, it's a particular isoform called KIR3.2 channel, self-administer more ethanol and fail to develop conditioned place preference for ethanol compared with wild type. Okay, so that means that they self-administer. That means they get intoxicated when they don't have that gene. So maybe that gene is a gatekeeper on some of the neuropathic effects. You see, understand the neuropathology and the pain and all of the conditions that lead to intoxication, morbidity, and death are the way for the body to respond to the toxin, not only by trying to degrade it, by trying to signal to the organ and the organism itself, right, at the central level, quit drinking the alcohol, right? So if you don't have this gene, then one of the ways to buffer or control or monitor that, in fact, gate it, is gone. So the results support a model in which ethanol may have lost its target in GERK knockout mice, thus failing to elicit behaviors associated with ethanol consumption. See? So receptor activation of GERK channels generates an outward slow inhibitory postsynaptic current, okay, which reduces neuronal activity. So you see, if you reduce that neuronal activity, right, you reduce that from this inwardly rectifying is going to then generate an outwardly, a response is always an outward flow of ions and ionotropic type of synaptic systems, then that normal flux, that normal control is overridden by ethanol ingestion. In fact, this paper showed that ethanol binds to the channel. Check it out. So it's called the ethanol toxicological paradox. Ethanol, of course, simple alcohol. I guess that's why they think it's a paradox, because gee, why a simple alcohol? It has two carbons, it has hydroxyl, yet dramatic and deleterious effects of mammalian physiology, involving behavioral, psychological dysregulation, and it does in many ways. We still don't know why ethanol and not a lot of other alcohols have the same effect, but they don't. Okay. Probably, I just think it's because ethanol is just ethanoic acid, right? Ethanoic acid is acetic acid, right? Ethanol is the alcohol of it. reduced form. I just think it's an essential metabolite, and that's why it has such a profound, perfunctory effect on metabolism. Whereas something with 3 carbons, 4 carbons, 5 carbons, you just don't synthesize that. You don't metabolize it, and you don't have entire grids like the TCA cycle that utilize those. At least only rare, like, for example, branched-chain amino acid degradation, stuff like that, where propionic acid, butyrate, and things like that get involved. And also an odd-chain fatty acid degradation. But I should stop talking about metabolism. Anyways, it was once thought that ethanol interacts non-specifically with membrane lipids. In fact, it does. So when it says, but new evidence, it does do that, okay? But new evidence suggests that ethanol also binds directly to membrane lipid and protein structures more specifically. That doesn't surprise me, okay? It has that OH group. And again, a while back in the fall, I gave this, I think, really cool lecture on the OH group and how fascinating it is in chemistry and also biology, of course. As I'm a biochemist, I had to bring that up. I recommend you go back and take a look at that. OH groups are just amazing. Anyway, counter to most such interactions of toxins, ethanol as a toxin, remember, ethanol exhibits relatively low potency. So look, you need millimolar amounts, okay? And appears to lack discrete biochemical specificity because, many alcohols might interact with these ion channels. Yet ethanol itself interacts with multiple ion channels. So you can get alcohols affecting these ion channels, binding and interacting with the proteins because they have OH groups. So they're alcohols. But ethanol has its own special repertoire and suite of channels which it specifically interacts with. So this study, this PNAS study, was alcohol tagging the pocket extensively with open GERK2. And it keeps, in fact, alcohol opens the GERK2 channel. Okay. So the ethanol tagging a single amino acid in the alcohol pocket, it's called, can induce or support global changes that lead to GERK channel opening. So there's a very specific interaction, drug ethanol, inwardly rectifying potassium channel protein GERK. Alcohol activation increases relative affinity of the phosphatidylinositol to GERK interaction. So once you open up that channel, you start turning on the signaling system, that is phosphatidylinositol phosphate signaling system, which goes into a whole other series of kinases, which then modifies and reinterprets what those cells are going to be doing. And that's because of the ingestion of the alcohol, right? So PIP2 association with GERK was shown to be essential for G beta gamma activation. The G coupled proteins, they have to break down and make the beta gamma form, and so these are same thing as you have adenylate cyclases, you have guanylate cyclases. It's part of the guanylate cyclase pathway. Okay, if you remember that. Results indicate that alcohol tagging the hydrophobic pocket of GERK channels increases the relative affinity for PIP2, leading to an increase in open channel probability. So they're starting to, basically what they're doing is they're dissecting, they're disambiguating, they're examining in fine molecular detail what ethanol is doing to this one particular channel. That's how one of the intoxicating modes. Now, ethanol works through membrane phospholipid. Ethanol activation of GERK2 channels slows PIP2 depletion. Okay, that suggests the enhancement of the relative affinity for PIP2 underlies this increase in open channel probability. So it really gets down to the molecular level, not just like tremendous changes, say, in transformation of one molecule to another, which people normally think about metabolism, but actually the rate of reaction, right? Or the slow decay or removal of PIP2 from the membrane. That's going to alter then the entire signal transduction cascade, but it's also going to leave it on. And you never want to do that in the system. You always want to have feedback control. So ethanol ingestion is not normal physiological, right? We don't make ethanol and utilize in our body to introduce all of these strange metabolic changes or central nervous system changes, let alone behavioral changes, right? There is no endoethanolic system like the endocannabinoid system. And by the way, I want to point out that I didn't in my cannabinoid talk, and I know we're not in cannabinoids right now, but endogenous cannabinoids don't look anything like exogenous plant-derived tetrahydrocannabinol or cannabidiol. Totally different molecules, right? So that's why THC ingestion isn't like turning on the endocannabinoid system, although it acts as a partial agonist to the receptor. Notice we're not talking about specific receptors for ethanol, but you can almost argue, in fact, if this was a test you were taking, let's say for your boards, and somebody said, is it true that ethanol has a specific protein it binds to? And you said no, you'd be wrong, because here's one, only one of many, actually. All right, so let's summarize this section of the research. Now, I'm not showing you data, because again, this is just an overview of ethanol and its terrible toxicity. Alcohol modulation of GERK2 channels regulates neuronal excitability in the brain's reward circuit. That's what GERK normally does. That's the dopaminergic pathway. This underlies, again, alcohol addiction. I didn't talk at all about that. The PNAS paper does. It's one of the reasons they did the research. But yeah, it's not just that you're slow release of PIP2 and you have this really elegant protein interaction with ethanol and the binding because of the size of ethanol, the two carbons, and where the hydroxyl is positioned in space-time. But also because, in fact, it underlies the whole reward pathway that makes people addicted to alcohol, the GERK, right? And then we notice that with the mice, right? You get rid of that and you remove that whole thing. They just drink alcohol, but the intoxication effect is simmered down. It's depressed, as is the addiction, right? Even though they freely take in the alcohol. So obviously something else is going on. Importantly, the canonical hydroxyl of alcohol was not essential for chemical-dependent activation of GERK. However, alcohol activation absolutely requires the membrane phospholipid signaling molecule PIP2, but not the receptor-activated G protein that is the guanylate cyclase. So the PIP2 is where you hone in on ethanol's effects. Chemical rules for hydrophobicity and size have significant implications for the development of chemical therapeutics that could occupy that pocket and could block access to alcohol, because we know it binds there, and that may reveal a new strategy for treating alcohol abuse and addiction. So in other words, come up with a drug that binds to that GERK2 site so it slows that PIP2. People don't get high from it, and they don't get addicted to it. That is, to what? To chronically ingested exogenous ethanol. All right. Okay, back to our paper in Alcohol, okay, the journal Alcohol, published in 2017, late, it was only a couple months ago. White matter myelin is a major component of the oligodendrocyte membrane. We saw that at the beginning. It functions to insulate nerve fibers and thereby support neurotransmission. Myelin is largely composed of lipids. Look at that. Love it as a lipid biochemist. Way to go as a system. 85% lipid. Excellent. And it includes these lipids, of course, cholesterol, sphingolipids, and the phosphoglycerolipids. Okay? Sphingolipids, like sphingomyelin, cerebrosides, and sulfatides, are located in the extracellular membrane leaflet and have a major role in myelin formation and maintenance and neuronal growth. That word, I left out when I was editing. Sphingolipids, together with cholesterol, form membrane microdomains. We talked about this in various VerEvMed lectures. Microdomains are how membranes get synthesized, how they fuse with other membranes, how they traffic and control packaging of proteins, for example, to leave the cell or to land into the plasma membrane and act as receptors or as voltage-gated channels. So once you start talking about lipid rafts, you're talking about intracellular signaling, which basically controls the entire fate and destiny of cells, right? And sphingolipids are so important. Again, I know I talked about that when I gave the general lecture on sphingolipids. Go back and listen to that. It's quite fascinating. And again, at the macro level, membrane regulating membrane fluidity, protein trafficking, and cylinder reduction. Of course, cholesterol is helping you control that fluidity because the ordering of the acyl chains. All right. Now, phosphoglycerolipids like PS and PE and PI are localized along the inner cytosolic leaflet exposed to the cell surface and function by regulating intracellular signaling membrane trafficking. So remember, lipids are so critical to understanding how biology works. Very few people are lipid biochemists. That's okay. Most people just don't have, let's just say, the effort. They don't want to put in the effort to become lipid biochemists. We'll leave it at that. But lipids are really important because there's so many different, thousands and thousands of different lipid classes, and they're organized into this macromolecular structure we call a membrane in a way that probably has a sequence in three-dimensional temporal signatures, which we just haven't been able to unravel yet. So unlike the simple macromolecules like DNA, RNA, and protein, lipids are just very, very difficult they're intractable still because we haven't developed a method yet to understand sequence of lipids and the rapid turnover in the membrane and how that affects this whole system. Anyway, progressive ethanol-mediated white matter atrophy and degeneration are associated with major alterations in myelin lipid composition. Big surprise, reflecting oligodendrocyte dysfunction, and that abstinence-induced recovery, look at here, and restoration of white matter integrity may at least in part normalize myelin lipid profiles. We don't really know what's going to do with the axon itself. For degeneration might be too late. So that means there's hope, and we know this. People who are alcoholics that quit drinking, the earlier they quit drinking in their life, the less they've had these chronic or abusive digestion of ethanol, the more they seem to be able to come back from it. And we know the person who's on long-term chronic ethanol abuse, that's where you start to see the Wernicke-Korsakoff syndrome. You basically see a person whose brain is no longer ever going to function normally again, right? No matter how much time they abstain from alcohol. So we know this. Now, this is a heat map. And, heat maps just basically show different color keys to show you how much is there, right? So this is just from that paper, and I wanted to show you. Here's a control, okay? And the control, you have a lot of red here. That means there's a lot of these, and these are all individual lipids. I'm sorry, this was actually a catch and grab using off a PDF file because I couldn't get this directly from the HTML document. But down here, all of these small lettering, the small wording here, all of these are individual lipid classes. So wow, a lot, right? Yeah, a lot of these are sterol lipids. You also have lactoglycerol lipids. So sphingolipids, sterol lipids, phosphoglycerol lipids, etc. are being represented here. Look at the control. A lot of this particular class of lipids, and then look at 3 weeks after ethanol ingestion, they go away, right? So remember Roy G. Biv, right? A whole lot in the red, less and less as you go through the Roy G. Biv color circuit. So then 8 weeks ethanol, look, more changes, right? The colors are getting more into the blue, from yellow to green, Roy G. Biv, right? Again, massive alterations in lipids, and then recovery, which is pretty extensive. This is an animal study. You don't get back to the control. You do not get back to the control. You do seem to look like stabilized here maybe, but there's still a continuation of alteration of all these lipid subclasses. All those lipid subclasses just in the myelin of the myelinated axon in certain parts of the brain and of the central nervous system of this mouse. So that means that even though in the short study, and again, these animals don't live long anyway, so this would be much longer if it was human, you do not completely recover these lipid classes. And if you don't have the correct lipid classes, you don't have the correct outer leaflet, inner leaflet, membrane lipid component so that you carefully and correctly traffic all the proteins and all the lipids and all of the endosomal systems, including those associated with the inflammatory response, you don't have normal physiology, even after recovery, because you've altered these lipids. So you have to come back eventually much longer, maybe. You start to see the resynthesis of the appropriate lipids, and then you'd maybe have a heat map that look more like this, but the study didn't go that far. Now, almost done. This paper from 2016, again, it's still a couple years old now. I don't want you to think that only lipids are involved, and you know that already, but I want you to just get the idea that ethanol ingestion, again, we're going to alter this entire acetyl-CoA pathway. You're going to make a lot more lipids, but also ethanol ingestion is going to modify all this other signaling phenomena. We already talked about PI signaling. Here it is working through the Wnt-catenin signaling. This is a major global signaling pathway that determines cell fate in terms basically of the whole process of cell cycle, but also which genes are expressed. These are basically transcription factor signaling systems, so they're major gatekeepers of what genes get expressed. And of course, the PI is the intracellular signaling. This is where lithium actually works, and when lithium is used in neuropsychiatry. But you also notice that methylated lipids, proteins, and DNA, this entire methylation pathway is altered by ethanol ingestion. And one of the main targets of that is your entire folic acid pathway. So another thing besides thiamine deficiency in alcoholics is folic acid deficiency. So that's another thing that they put in the catheter when you're trying to give essential fluids back to an alcoholic during rehabilitation, right, during detox. So, and that's because folic acid synthesis becomes corrupted. Now, when you corrupt folic acid synthesis, look what you corrupt. Nucleic acid synthesis. So obviously, DNA, RNA, and all the nucleotides, that is, for example, the ribonucleotides like ATP, GTP, CTP, that are utilized in intermediary metabolism, all of that synthesis is also corrupted by ethanol ingestion, directly by impacting on folic acid metabolism. Likewise, since folic acid is associated with methylation pathways, all the methylation using S-adenosine with thionine, which occurs, for example, during chromatin remodeling. So you have, again, the histone code, which is modified by methyl groups on particular histones, the cytosine residues, which are involved in epigenetic signatures in DNA, which controls gene expression. All of that is going to be impacted by ethanol ingestion. So that the entire global genome can be altered irreversibly and reversibly because of epigenetics, but certainly passed on to multiple generations because of epigenetics, and also because of point mutations that occur because of reactive oxygen, because of the ingestion of ethanol. That's why, of course, it's such a teratogenic substance. That's why ethanol is also teratogenic. It's why pregnant women don't drink alcohol. Here's a dialectic then. It's a very, very general dialectic, purposely. Ethyl alcohol has been consumed by humans since earliest recorded time, going way back at least probably to the Assyrians. We've been able to see discussion of fermenting of drink, which obviously means that they were making something that ethyl alcohol being made. Ethyl alcohol is toxic to the central nervous system, the peripheral nervous system. Certainly, I hope that I've convinced you of that just briefly today, but just take a look at the literature and I think it won't be any difficulty convincing you of that. Via multiple mechanisms, as I've been saying, membrane disruption, neuronal and glial structural malformations, such as we talked about arborization on the last talk, metabolic dysregulation, just think of the NADH and NAD couple, inflammation, the microglial activation, but also, of course, when you activate microglia, you also will then activate circulating macrophages, and that then can induce an entire T cell response, which, of course, is wide open. In fact, chronic ethanol ingestion does have a very strong pro-inflammatory response, causing autoimmune responses. Again, that's another entire topic. And that, of course, if it's in the brain, that can cause wreak havoc and cause neurodegeneration. And that's what happens. Consumption of ethanol. So here, okay, there's the synthesis, antithesis. Here's the synthesis, thesis, antithesis, synthesis. Consumption of ethyl alcohol must impart some perceived benefit in that it otherwise is chosen over sober judgment. Again, that's my opinion on the subject, but I call it a dialectic because I think it's actually a very good sober judgment. Ethyl alcohol, even small amounts of it being ingested, are causing damage to the central nervous system, peripheral nervous system, the immune system, and all the solid organs, including, for example, the heart and the liver, as we all know. All right. So we're going to finish there on that unhappy note by saying that alcohol, basically, that is ethyl alcohol, is a toxin. And so please use it very sparingly, if at all. Total absence of alcohol will not cause any disruption of an otherwise very sunny romantic life, I'm sure. Okay, so now we're saying goodbye. Again, this is the Continental Divide in Colorado, place I love. There's a Facebook which has all of my VerEvMed lectures in it. Go and check them out. Again, I'm cataloging them and putting them into the VerEvMed site. It's just slow because I don't have help. That's me, Dan Guerra, and that's my individual email address. Contact me if you have any interest in pursuing what I talked about today or any of my other lectures, or just want to branch out in a totally different area. The email address of our company is info@verevmed.com. Please reach us there. We will respond to you quickly from there, and there's our web page. And remember, we are scientists verifying published evidence in medical bioscience. That's why we formed our company. All right, so we're ending the show there. And all I have to do is do my sign off, which is goodbye for now.